I don't think it's an exaggeration to say that if you understand this image as well as this image and how the two of them go together, that you're going to do fine in your physiology course. And that is because these structures here on this image, I mean, this is how cells work, more or less. I mean, obviously you have the internal mechanisms, because what we're looking at here is the cell membrane. And the cell membrane is incredibly important. And without it, I mean, literally nothing works. I mean, it's kind of like, do you have a house if you don't have walls and a roof and a floor, right? Obviously, the cell membrane is what creates the boundaries for the cell. And so there's a lot that is happening here. And in today's video, we're going to be using this study unit. This is a physiology study unit from Kenhub to explore the cell membrane. Now, this is a premium feature, but we're going to go ahead and leave a link to another study unit for you to go ahead and check out. So that one is going to be on ReflexArc. You'll be able to see everything that we pack into these amazing study units. And plus, when you sign up for Kenhub for free, you're going to get access to our thousands of images, thousands of articles, all for free. So be sure to check that link down in the description below. So what I want to do is hit this little magnifying glass so we can better see this right here. Because like I said, this is the cell membrane. And so the cell membrane or plasma membrane, it's pretty much interchangeable, like I was saying, is the boundary of the cell. And so we need to understand as much as we possibly can about this. But interestingly, it's actually not that complicated as long as you start grouping things together. Oftentimes I'll see students just get overwhelmed as they're looking at it, which is fair. I mean, there's quite a bit going on, but when you know what to look for, it's going to get a lot easier. And so it all starts with this right here called the phospholipid. So if you look closely, you're going to see that we have like this gray sphere and then we have these two tails. Well, imagine we pull one of those out, and that's exactly what you see here. So the entirety of it is called a phospholipid, but we can also further separate it into two distinct portions. So we have what's called the hydrophilic head, and then we have what's called the hydrophobic tail. Now, when we're talking about, so let's actually say this. So hydrophobic, I think most people understand phobia, meaning fear. So quite literally, hydrophobic tail means that it fears water. Now, is it like literally fearing it? No, but it's basically, it doesn't want to interact with water. Now, philic, if we actually look at what phil means, it means attraction. So hydrophilic heads are attracted to water. So what's interesting about this is because this is the perfect molecule to actually make a cell or plasma membrane out of. Because you can see all of them lined up here. What'll happen is just by like putting them together, because you have to understand, you see where it says extracellular fluid? And then in here you can see where it says cytosol. So these are fluids. These are water. So that means there is water on the outside of the cell and water on the inside of the cell. And so by placing something that has one end that is attracted to water and the other end that repels water, it's spontaneously going to organize itself in such a way that you have created a really effective barrier. Now we're going to see this is a selective barrier, right? Because this is going to be semi-permeable, and this is extraordinarily important to understand about the plasma membrane, is that some things can just waltz on through, almost like a ghost, just going through a wall. And other things need to use a door, or they need to have some other kind of mechanism that is going to bring them through. But this is a very effective barrier. Right. So, it's really, really cool how it just kind of like organizes itself that way, because, you can see like the two tails and two tails that way. In fact, we call this a bilayer. So this is a lipid, a phospholipid bilayer, because you have one on this side, one on this side, and it's going to do a really good job of keeping the water, which inside the cell is called a cytosol. It's more than just water, but just for now, just think, keeps the water that's on the inside, and the water that's on the outside. It's perfect. It's awesome. Absolutely great. But there's other things going on in this cell membrane. So the first thing I want to kind of bring your attention to is going to be this peripheral protein here. Now there's many different types of peripheral protein. So again, this is what we're doing here in this image, is trying to simplify things as much as we can into groups so that you can better understand what they're going to be doing. Now, the peripheral protein, for one, it's actually going to help just kind of maintain the shape of this cell. So what will be like attached in here is going to be parts of like the cytoskeleton. So those are proteins that are going to be in the inside, and so that's actually going to help keep the cell shape, right? This is essential to keeping the cellular shape, because without it, I mean, you have to ask yourself, like, Imagine if like all the cytoskeleton, which is literally like scaffolding, it's just like all these proteins going in these different directions. But if those were not physically attached to the cell or plasma membrane, it's like, how is that working, right? It's kind of like not having a load-bearing wall or something like that. It's not going to be good for structure. But there's also some enzymatic activity here. So can be enzymes. There's all sorts of multiple functions that we're going to see for a lot of these. But peripheral proteins, you can see this is on the inside of the cell. So this is intracellular, or at least, and then it's like entering into the physical membrane itself. Now, then we can see a globular protein. So globular proteins, these can do a lot of different things. My favorite globular protein to talk about is actually hemoglobin. So hemoglobin, which is you're going to find on a red blood cell, that is there to kind of like attach to oxygen, right? So it's going to grab the oxygen that you breathe in, and then it's going to kind of shuttle it around, and then that oxygen we're going to see is going to diffuse into cells. But that is one example of a globular protein. So globular proteins, these can attach to a variety of different things, kind of like transporters. They can even move things in and out of the cell if they need to. Kind of depends on what it is. But you could kind of think of a globular protein as going to be attaching to something more often than not. Then over here we have cholesterol. So cholesterol gets a bad rap. Like I get it. Like when we say like, oh, you have a bad cholesterol or high cholesterol, you know, we're really talking about very specific things. And that's not really the type of video we're doing here. We're like talking about like HDL versus LDL versus VLDL and all those different types of cholesterol. We're just going to say broadly, what is cholesterol doing for the plasma membrane? And what it does is it helps break up the phospholipids. So basically it's going to make the shape so it's not too firm, right? Without the cholesterol, like basically temperature can really dramatically impact the shape and the fluidity of the cell membrane. So if it's too hot, then what'll happen is the membrane just wants to kind of like, you could almost think of it like melting. It's not really, but I want you to think it becomes more fluid and just, you know, mobile. And if it's too cold, then it becomes too rigid. Well, what's going to happen is the cholesterol is going to be positioned in varying places within the plasma membrane, and it's going to allow the cell to still function normally despite those temperature changes. So cholesterol is absolutely essential to the shape of a cell and to the health of that plasma membrane. So, you know, again, this is why, you know, nutrition and just as you hear it on the internet is not really a great way to go about it, right? Cholesterol is not bad. Cholesterol is essential. It's just one of those things that you want to, you want to have the right types, and you want to have even the ones that are not the right types. You can have those. You just want to have them in moderation. All right. So then we have glycolipids. So glycolipids, these things, they could do a lot of different things. So for one, I think the one that I want to talk about here is this one actually can help identify, like the cell, right? I mean, that the globular protein also does the exact same thing. So again, another function of a globular protein would be like the MHC complex. So it's basically, I think of both of these, we'll put it this way, both of these almost like name tags, because you have to think to yourself, how is it that a cell knows who that cell is? Right? Like all my cells have just in name tags. Most of those name tags are going to be globular proteins, but there's also other kind of identifying name tags. So imagine like you have, like almost like a key card for your place of work. Not only does it say who you are, but also gives you like access, tells you all sorts of different places you can go, or your level in the company, so on and so forth. Like a glycolipid, this could be like blood types. So if you've ever heard of like, I'm A positive, right? So A positive, you have A negative, right? You have AB, like you have these different blood types. Those are going to be glycolipids. They're just little, glucose and fat-based molecules that are going to help kind of determine various things about the blood cell, right? There's a lot of different ways that glycolipids can actually manifest in cells, but that is just a real easy example. You can just think of like blood types and those types of things. All right. So then we look at channel pore and channel protein. So, depending on whatever textbook you're looking at, you might see this be worded differently because there is a difference between certain channels versus pores. They get to be pretty nuanced. What I want you to just kind of understand is these are going to be doors. All right, so imagine like you have a physical door And then you have the opening to the door. So it's like the door frame and the actual like door that closes. And then when you open the door, there's obviously a path you can go through. I think that's going to be the easiest way to kind of just think about this right now, but there is a lot of nuance into how these work with like voltage gated and there's all sorts of nuance to it. But just think like there's going to be an opening that would be the pore, and then you're actually going to have a protein that forms the door frame and the physical door itself. And these are going to be uniquely shaped, right? These are these are going to be uniquely shaped to where they're going to be shaped to the exact type of like ion that we're going to see going through it. It gets to be pretty cool. I always, when I'm teaching this in the classroom, I pretend that like I am I'm like an ion trying to go through this one of these doors. Well, that door would have to be Justin shaped, and I'd have to kind of fit through it in that specific way. So they get to be really, really nuanced. All right, then you have what are called the integral proteins. And so these ones will actually span the entire length of the membrane. So if you look here, like the peripheral protein doesn't, the globular protein doesn't, but the integral protein, these ones are actually going to span the entire length of it. And they themselves are hydrophobic as well. And so that actually mixes with the hydrophobic of the nature of the phospholipid. And this is also going to be attached to things. This can attach to the cytoskeleton. This can attach even to a peripheral protein. So like you could picture like a peripheral protein being attached to an integral protein on the other side. They can help move things through if they need to. There's several different functions that are going to happen with integral proteins. And another thing I just remembered right now, like another globular protein might be an integrin. That's why I was thinking about that. An integrin is kind of like how cells can dock to each other and kind of attach. So then they're not, they're actually physically connected to each other and not just hoping that, they, can you imagine if cells just kind of like your skin cells, for example, just had to hope that they just stayed next to each other despite all the movement and friction and all that kind of stuff. So there's a lot of diverse functions here. And then up here, by the way, this is trying to just show you a different view of one of these protein channels. That's all we're seeing there. And then you can see another cholesterol here. So like there's a lot going on here. And again, this is a very simplified version of it. But if you even just understand this, that you basically have doorways, you have name tags, you have support structures that help create like, mobility and fluidity, but also retaining strength within the wall. If you have, docking stations and, like you're going to pretty much understand the overall functions of the things that are located inside of the plasma membrane. But what I wanted to look at now is this, right? So what are the things that can get through it? Because remember, as I was saying, it's a semi-permeable barrier, meaning that some things can just waltz right through. And that's what you see here on the bottom. Other things, if they want in or out, they're going to have to take advantage of, say, like a pore or a globular protein or a peripheral protein or an integral protein. Like there's going to be rules to how you get in. And it's no different than, say, like if you have like a key to a house, right? Or a biometric scanner at work or something along those lines. I don't know where you work, but you know what I'm saying? Like there's going to be like a security system for things to get in and out. And that's very important because if the name of the game here, besides just optimizing function and being healthy, is also preventing pathogens from taking advantage of these systems. So you have to have your two-factor authentication and authentications and all that kind of stuff. All right. So first up, if you look at gases, this is, the easy examples here are going to be like oxygen and carbon dioxide. You have to understand that cells produce carbon dioxide. So the cell needs to be able to get rid of it. And then also, the cells need oxygen. So imagine this is the outside of the cell. You're going to have a red blood cell coming by. Attached to one of its globular proteins, the hemoglobin protein is going to be oxygen. It is then going to give the oxygen, and the oxygen is going to diffuse, which means basically it's just going to go through. Again, think of it almost like a ghost, which is really easy when you think about a gas. They can just go right through this barrier. They are not concerned about, the hydrophilic and hydrophobic portions. They just go right through. And so then you're going to have an exchange. The carbon dioxide that the cell made is going to be given over here, and then the oxygen that it needs is going to come out through here, and so on and so forth. So That is all happening through diffusion. That's just following gradients, right? There's less oxygen in here than there is here, so the oxygen naturally wants to go this way, the carbon dioxide wants to go this way. Steroid hormones. So steroids are actually pretty massive. The thing to understand, though, is that they're lipid-based. And so when they're lipid-based and you have a lipid bilayer here, they can kind of just do what they can just kind of like whoop, go right through. So that's not so much about a size thing as much as it's just about its molecular makeup. So steroids, just that they're going to kind of go through. And then water we have to be really careful with, because it's not like water can just go through as much as it wants, because remember, we just said this is hydrophobic. Since it's hydrophobic, that means it's going to be able to dramatically limit how much water gets through. But it's not like there aren't cracks in here that the water can't slip through. And so H2O, right, the molecule is capable of going naturally through here. But there's also, if we go back, we're talking about pores, like there's something called like an aquaporin. An aquaporin is going to be a specific opening for water to just kind of go in and out of the cell. So there's also going to be those, right? So we have the water has the ability to naturally just go through, like through osmosis. And then it's also going to go through aquaporins and things like that. All right, so then we look at these. Now, proteins just tend to be too massive, right? It's just way too massive to be able to fit through here. And so proteins are going to require some other kind of protein to get them in or out of the cell, because it's also possible that the cell produced a protein and it wants to get rid of it. That protein is going to have some form of mechanism to go through. Now, typically that's going to be like vesicular transport too is another option that we haven't talked about. Just understand the cell can even produce like a little package of phospholipids that'll like attach and become part of the cell membrane and push the protein out. Right. So there's other options too, and really creative options. Sugars are another thing that you're not going to be able to just naturally go through. Like glucose is going to need some kind of carrier molecule to help get it through. And glucose is essential, right? Without glucose, the cell can't even produce energy in the form of ATP. So it's amazing. Again, these things are necessary. It's just, we also want to be very careful about who we let in. And then this last one here, we're talking about ions. So you can see like sodium, potassium, chloride. These are going to be, essential for how the cells work, right? Some cells more so than others, but essential for all the cells. The easy example is going to be neurons, right? So neurons are going to use the charge, right? So you have like positive, negative, to actually change the voltage inside and outside of the neuron. So imagine, basically how a neuron would work. The nitty gritty of it is on the inside, that environment at rest is less positive than outside of the cell. And then by shuttling in sodium ions into it, you increase that charge. And if you reach a certain threshold, you trigger an action potential, and then all of a sudden that neuron can fire. So by clever usage of charged ions, right? Well, an ion is an atom with a charge, but by the clever usage of an ion, you can actually get work done. So you don't want to just let it in and out as much as you want, because that would defeat the entire purpose. Instead, you want to be very specific about when it gets in and when it gets out, so then you can actually do work with those charges. It's absolutely brilliant. Absolutely brilliant. So this is it. This right here, again, there's tons of nuance. There's tons of nuance to all of this. There's different types and subtypes and all sorts of things. It's just, if you understand what we're just talking about here, I think you are golden for your physiology course, because then you can just start asking yourself, okay, well, what type of globular protein, right? What type of pore? And once you start doing that, things are going to make a lot more sense. All right. So again, this was part of a larger physiology study unit. These things are amazing. They are just gigantic lessons. As you can see, there's so much going on here. It's even talking about, the resting membrane potential at negative 70 millivolts. There's so much that we pack into these lessons. Again, these are premium features, but at the same time, Kenhub offers so much for free, so be sure to check it out. We're going to go ahead and leave a link down in the description below to a separate study unit on reflex arcs. It's really cool. It's kind of like when the doctor hits your knee with a mallet and then it goes up like this. You're going to learn how reflexes work. It is really, really fascinating. So be sure to check that out. And while you're down there, if you enjoyed this video, you found it useful, go ahead and hit the like, subscribe button, leave us a comment. All that stuff is extraordinarily useful. But thanks for hanging out with me. I genuinely appreciate it, and I will see you in the next video.